Now, our next speaker is Dr. Harpreet Kaur. Harpreet Kaur is a scientist from the New Zealand Institute of Plant and Food Research Limited from New Zealand, and she's doing some splendid research in related to food and processing of food. And today she's going to talk about correction of temperature variation. This is a problem that we very often face when we are analyzing the data. So she's going to talk how she did that in order to predict soluble solids content in the Kiwifruit juice. Of course, she used NIR, spectroscopy, and aquaphotomics. And Harpreet is actually today with us, so she can answer the questions. She can answer the questions after the video finishes. So, Lawrence, can we now have a video of Harpreet? Dr. Kaur. Okay. Thank you, Lawrence.
Hello, everyone. My name is Harpreet Kaur. I'm a research scientist at Plant and Food Research, New Zealand. And in this presentation, I will talk about my paper that has been published in the Molecules Journal from MDPI, and a special edition which is called Aquaphotomics, Exploring Water Molecular Systems in Nature. For this paper, I would like to acknowledge my supervisors, and many thanks to Professor Sinkova and Gelena for inviting me to write this paper. And many thanks to Tsukino Shizuku Foundation for providing me funds to publish this paper. So in this paper, I will talk about correction of temperature variation with independent water samples to predict soluble solid content of kiwifruit juice using NIR spectroscopy. Just making it short and simple, I will talk about how to get rid of temperature effects using independent water samples. As we know that when a calibration model is made at a particular temperature, for example, I'm taking a temperature model developed at a particular temperature. So when a model is developed, and when we want to measure new samples at a different temperature, so what usually happens? We get errors, we get incorrect readings because of this bias problem. So can aquaphotomics, in case of fruit and fruit juice samples, can aquaphotomics help us to understand and get rid of this bias problem? Can we use independent water samples to get rid of this temperature effects? This is the main highlight of this paper. So the content of this presentation is I'll talk about the quality assessment of fruit, NIR spectra and temperature variation in case of fruit spectrum, need of aquaphotomics, experiments in aquagrams. The main two techniques which I have talked in this paper is extended multiple scatter correction and external parameter orthogonalisation, some results and conclusion. So in New Zealand kiwifruit industry, the dry matter and soluble solid contents are two important parameters. These are the internal fruit quality parameters that judge whether the fruit is ready to be harvested and how good the taste of the fruit is. These techniques are destructive in nature. Dry matter, it takes 24 hours to get the actual dry matter content, and SCC is by squeezing the juice, cutting the fruit, squeezing the juice onto the refractometer. So they are time-consuming, destructive methods. So here comes near-infrared spectroscopy that simplifies everything. It is fast, non-destructive. So when a fruit is measured at, the spectra of fluid is measured at different temperatures. So what we get, so in the second overtone region of water at 970 nanometers, you can see there is a change in the absorbance intensity, as well as there's a wavelength drift. So this wavelength shift around the second overtone of water is due to temperature. And it's only at, it happens only at the water band. So when a model is made at one particular temperature and you measure a sample at a different temperature, there is a bias, and this bias gives incorrect readings. So because of this shift on the water peak, we thought about why not to study aquaphotomics, because aquaphotomics talks about perturbation in an aqueous bioaqueous system. So in our case, the main perturbation was temperature. So we do normal NIR spectroscopy, we get the spectral database. And from the spectral database, we can find out the activated water bands. From the activated water bands, we can get how the water spectral pattern changes with a perturbation. In our case, in my case, it's temperature. So why I took fruit juice? To better understand how aquaphotomics can work in the first overtone region, that is from 1300 to 1600. And also I have studied this same aquaphotomics methodology in the second overtone region from 870 nanometer to 1100 nanometer. We wanted to understand the water structure. So that's why I took the fruit juice sample. And why water samples? Because I wanted to see whether I can use an independent water sample that can act as a correcting matrix or an interfering matrix that can correct my fruit juice spectra and give me a model that is not dependent upon temperature variation. So that was the main purpose. And I wanted to see how aquaphotomics can help me to develop robust calibration model against temperature. So in the experiment, we took 100 kiwifruit samples. These are the goal 3 fruit, kiwifruit. The juice was extracted and collected in Eppendorf tubes. They were centrifuged and filtered. Afterwards, the juice was measured to get the soluble solid content using a digital refractometer. Using a Bruker FT-NIR spectrometer, the juice was measured at 3 different temperatures, 20 degrees, 25 degrees, and 30 degrees Celsius, using 2 different cuvettes. The first cuvette was 1 millimeter path length cell that gave accurate absorbance around 1300 to 1600 nanometer region, and 10 millimeter path length cell longer in the short wavelength NIR region, that is in the 870 to 1100 nanometer region. Independent water samples were also measured at 3 different temperatures. I measured the independent water sample, the molecule water, at the beginning of the experiment, then at the middle, and last one at the end of the experiment. Talking about the absorbance spectra of juice, as you can see in the first diagram, in the first overtone region from 1300 to 1600 nanometer, with an increase in temperature, there is a lateral shift in the absorbance peak. 1444 nanometer is the isobastic point. However, in the second overtone region, from 870 to 1100 nanometer region, there was a vertical shift in the absorbance peak With increase in temperature, the peak intensity increases. There's a slight lateral shift, but it was more vertical shift. The isobastic point was around 994 nanometers. Water matrix coordinates were used from Professor Sinkova and Munchin paper, where they have found a 12 water bands in the first overtone region, which is in 1300 to 1600 nanometer region. So as you can see, these are the water matrix coordinates that defines the water species. And using the enharmonic oscillator model, I found 12 water species in the second overtone region of water. So how to identify the water matrix coordinate? Which water species is activated with temperature perturbation? So I took the spectra at 3 different temperatures, did the principal component analysis, and using the first loading, principal component loading, I have found out this pattern, this curve, which gives a peak and a dip at 1414 nanometer and 1494 nanometer. The first graph is for first overtone region of ovate stretch of water. Similarly, in the second overtone region of water, the two peaks were 963 nanometer and 1027 nanometer. So these peaks correspond to particular water species. So in the first overtone region, it is C5 and C11. In second, it's C6 and C12. C5 relates to free water species. C11 relates to S4 species, water molecule having 4 hydrogen bonds. C6 is related to white water hydration, and C12 is strongly bounded water. I was expecting to get similar species in both first overtone region and second overtone region of water. However, the results shows some difference. After finding the water matrix coordinates, aquagrams were plotted, and you can see with increase in temperature, the free water species increases. Since there was a difference in the water matrix coordinates, the peak and the dip between the first and the second overtone region, you can see the aquagrams were slightly shifted, but there is a trend from bound to free water species. So with the increase in temperature of kiwifruit juice samples, there is an increase in free water species. Waters was also measured at 3 different temperatures. And using those water samples, I found out what is the correction matrix that can be used as an independent matrix that can be used to correct the kiwifruit juice samples at 3 different temperatures. So the spectra, the calibration spectra, the prediction spectra was corrected. This is the original spectra. The coefficients were found using multiple linear regression, and this I interference spectrum was taken from water. So you can see the principal component loading of water, the PC1, that looks very similar to the kiwifruit juice samples. Similar for second overtone region. So what I have found then, using EMSC technique, when a calibration model was made at 30 degrees Celsius and validation was done at 20 degrees Celsius, in the first overtone region of water, there was huge reduction in bias. That is from 0.77 to 0.1 degree Brix. Using the EPO, external parameter orthogonalization correction technique, which is basically done using the difference spectra. The principal component analysis was done on difference spectra, and difference spectra is found by taking difference of the spectrum at various temperature from the lowest temperature spectra. So when EPO was used, you can get more details in my paper as well. So calibration was done at 30, validation at 20 degrees Celsius. Using in the second overtone region of water, the EPO technique reduced bias from 0.5 to 0.04 degree Brix. So in the conclusion, I would like to say that aquaphotomics has helped to understand different water species, how with temperature perturbation, what water species gets activated, and what is the main trend. Around the water band in the first and second overtone region of water, in the first overtone region of water, that is from 1300 to 1600 nanometer region, there was a lateral shift when the temperature was increasing, whereas in the second overtone region of water, it was more a vertical shift. Using EMSC and EPO preprocessing techniques with independent water spectra, it has reduced bias to insignificant levels. So it means that using an independent water spectra, we don't need to measure the same sample at different temperatures. We can use sample at 20 degrees Celsius to measure, to predict samples at 30 degrees Celsius. So it saves time. We only need to measure water samples at different temperature. It allows measurement at different temperatures to the calibration set. Thank you very much. Hope you like my presentation. For any questions, please email me at harpreet.kaur@plantandfood.co.nz. Thank you very much.
Thank you very much, Harpreet. Harpreet, are you here with us?
Yes, I'm here.
Yes. Thank you. Wonderful lecture. And oh my God, I so love this way of presenting slowly. And, you know, like, I really have to learn more how to present. Anyway, I just wanted, so we have one question, but before I give you to answer to Pierre, I just wanted to say that this paper is not only about the kiwifruit juice, and this is really such a fundamental work that you did. And I would like to just also mention that you have much more work behind you, and a lot of very, very good information can be found in your PhD thesis, which is also available online. And I'm using it a lot, especially for this second overtone. So I just wanted to say that and to comment, because not many studies are done on second overtone, and it's really valuable. So thank you for this. And I really hope that other people are seeing what I just said, that this is not only about one thing, about one fruit or just one system. This is really fundamental, important. I mean, you're showing actually that the second overtone and the first overtone are giving different information. So they're influenced by different things, and of course that's due to the light that is used as a probe. Now, I'm sorry, I wanted just to comment about, I wanted to emphasize this importance of your lecture. Now, if you can see, there is a question from Pierre in chat box. So I would like if you can answer.
Yes, so regarding the whole fruit, we have done an experiment for whole fruit as well at different temperatures. And as I said, in the second overtone region, there were two water species. One is at like 963 nanometer and another one at 1027 nanometers. So the same species get activated. So this was the main finding after doing experiment with kiwifruit juice samples. So still we are in process how we can use the EMSC and EPO methods for the whole intact fruit measurement, because with kiwifruit juice sample, it's quite easy to use 1 millimeter Parkland cell or 10 millimeter Parkland cell, the fixed Parkland cell, and apply the method of EMSC and EPO. But with kiwifruit, intact kiwifruit, it is quite hard because the penetration depth, it changes when you pass light inside the fruit. It gets like scattering effects. So we are still dealing with that problem. But yes, the Aquaphotomics technique, it works on intact kiwifruit as well. We get the same bands in the second overtone region of water for whole kiwifruit. So it works.
Okay, thank you.
Pierre, are you satisfied?
Yeah.
Yes.
Yeah, I'm satisfied. Thank you. I was just wondering, because when we tried our cherry tomato tests, we saw some differences between an entire fruit measured within the Aquaphotomics system, or let's say in the NER spectroscopic device, and one that is cut open. And I wonder if, and this is just a hypothesis, I wonder if we manipulate it in terms of destroying the integrity of the fruit, that we induce decoherence in the aqueous phase. If you look at the quantum electrodynamic picture, it represents, or it means, or it implies, if you want, that the solid phase, the biomolecules that suddenly gets ripped off during the cutting, or if you just say you extract liquid phase, the solid phase that's missing changes the coherent fraction within the liquid phase. So we no longer have the attachment or alignment of a coherent fraction attached to the biomolecular structure because it's missing, because we have extracted that. And this could change the absorption bands in the spectrograms. But this is just a hypothesis, so this needs to be elaborated.
I understand totally what you're why you're asking this, and however, in Harpreet's problem was she showed she had a specific and problem to just predict soluble solid content, and in the I don't think that this matters that much. However, if you wanted to predict whether something is going to develop a disease, or if something can develop into a whole, like nice plant or not, then doing things like, invading the structure of your biological system has absolutely impact. It depends on the purpose, on the objective of your study.
All right. Okay. No, it's just.
I think that's. What do you think, Harpreet?
Yeah, I think it depends upon the objective of the study. So in my case, it was. I haven't gone, deeper into the part looking at the coherence or but still I have compared the intact fruit with the juice measurement. So
that's fine, because it gives me confirmation. Thank you.
Cool. But the techniques that you developed, that you tested, the EMSC and the EPO, this is very, very helpful to just, for example, to do before we do aquagrams. And this temperature effect is, I know in our labs, that is the problem which, gives us the worst headache. There are aspects of temperature influence that we like and that we are using. But sometimes when we just want to present something very, visually simplified in Aquagram, then this temperature bias can be really, really, really bad. So this is an interesting thing. I'm always on the lookout. I know that SMB doesn't work very well.
No.
It can distort the spectra. EMSC is my favorite, basically. Yeah. And in your case, what I really liked that you're using pure water. You're using water as a standard, the way to calibrate basically your.
So that was the interesting finding after studying aquaphotomics, because everything because it's a bio-aqua system, so fruit, it's happening because of water. So why not to use water as a standard or a standard sample to do correction. Why should we measure fruit samples at different temperature, which is time consuming?
Exactly. So that's beautiful. That's like using a water as a thermometer. And we actually can use water also as a clock to measure time. Anyway, I'm sorry, this was very inspiring, so I took a little bit more time to comment. Harpreet, thank you very much for this and for writing this paper, and which is very nice. And I like the next webinar we'll also have your colleague and you, I guess, who worked on a very, very nice exotic topic. So we will see each other again in October, and I hope you will again be you know, in a position to publish with us and work on aquaphotomics in future. So thank you for accepting to do this paper and to give a talk today and be with us today. So just a little bit of applause.
Thank you very much.
If anyone wants, I gave the link to the paper of Dr. Kaur, and we can now go to our third speaker today.